During the most extreme winter in Earth's history, when glaciers smothered the oceans and ice stretched from pole to pole, our planet became a frozen sphere drifting in silence. Yet life refused to disappear. In the darkness beneath the ice, in vents of superheated water, in thin films of brine, and in microbial fortresses carved into the cold, extraordinary organisms endured conditions that should have extinguished them. These resilient survivors, microbes, early animals, and ancient cellular architects became the foundation for all future life on Earth. Their persistence during Snowball Earth is not just a story of survival, but the reason complex life, ecosystems, and eventually humanity were ever able to exist at all. Cyanobacteria, the pioneers of light. Imagine a world locked in ice, stretching from pole to pole, where sunlight struggles to penetrate a thick, frozen ocean surface. The temperature is near freezing, nutrients are scarce, and life as we know it seems impossible. Yet, in this seemingly inhospitable environment, one group of organisms defies all odds. Cyanobacteria, the tiny, photosynthetic pioneers, whose resilience and ingenuity helped preserve life during Snowball Earth. These microscopic powerhouses were more than simple cells. They were architects of survival, oxygen producers, and the foundation for the ecosystems that would one day flourish as the ice began to recede. Cyanobacteria are remarkable not only for their ability to photosynthesize, but also for their extraordinary adaptability. Unlike higher plants, which rely on abundant light and stable conditions, cyanobacteria can survive under extreme stress, adjusting their metabolism, cellular structures, and pigmentation to optimize energy capture. During the Cryogenian period, when Snowball Earth gripped the planet, these organisms exploited every thin layer of light that penetrated the ice, whether through cracks, fissures, or thinner ice sheets, often forming dense mats on the underside of frozen surfaces. Each mat, shimmering faintly with chlorophyll and other pigments, became a microcosm of energy, sustaining not only the cyanobacteria themselves, but also a host of other microorganisms that relied on them for oxygen and organic carbon. Their survival was further enhanced by the evolution of protective strategies. Cyanobacteria produce extracellular polymeric substances, a kind of sticky, gelatinous matrix that envelops colonies and mats. This substance provided insulation against freezing, reduced water loss, and stabilized delicate communities against turbulent meltwater currents. In effect, cyanobacteria created their own microenvironments, small pockets of life that resisted the harshness of global ice. These biofilms not only protected individual cells, but also facilitated nutrient recycling, capturing minerals, and trapping organic matter that might otherwise be lost in the extreme currents beneath the ice. The physiological flexibility of cyanobacteria was another key to their endurance. Many species could adjust their photosynthetic machinery to capture even the faintest light, shifting pigment composition to optimize energy absorption at different wavelengths. Some could slow their metabolism almost to a standstill during prolonged darkness, entering a form of dormancy until light levels increased. Others developed filamentous or colonial structures that allowed them to float just beneath the ice or adhere to surfaces in meltwater channels, maximizing access to both light and nutrients. In this way, cyanobacteria became highly efficient survivors, exploiting every microhabitat available in a frozen world. Equally important was their role in shaping ecosystems. Cyanobacteria were primary producers, the first link in a fragile and minimalist food web. They converted inorganic carbon into organic matter, fueling bacteria, protists, and other microorganisms that survived beneath the ice. Their oxygen production, though limited by low light, began the slow process of oxygenating isolated pockets of water, setting the stage for future multicellular life. In essence, cyanobacteria were both life preservers and ecosystem engineers, creating conditions that allowed other organisms to endure alongside them. Modern analogues of these resilient pioneers provide a window into their ancient strategies. Cyanobacteria thrive today in Antarctic ice, frozen lakes, and high-altitude glaciers, enduring sub-zero temperatures, low light, and limited nutrients. They often form mats that are remarkably similar to cryogenian biofilms, illustrating that the survival strategies perfected millions of years ago are still effective today. These living fossils suggest that cyanobacteria's success during Snowball Earth was not accidental. It was the result of millions of years of adaptation to extreme conditions. Cyanobacteria also employed symbiotic relationships that enhanced survival. Some formed partnerships with fungi, early protists, or even other cyanobacteria, exchanging nutrients or stabilizing shared biofilms. These collaborations were vital, especially in a world where resources were scarce and environmental conditions fluctuated unpredictably. By forming dense, cooperative communities, cyanobacteria amplified their collective resilience, ensuring that even as glaciers shifted and ice sheets thickened, pockets of life persisted. In addition to their ecological role, cyanobacteria had profound evolutionary significance. By maintaining photosynthesis under extreme conditions, they preserved the pathways for energy capture that would later be essential for the rise of algae, plants, and eventually animals. They were the first to demonstrate that life could not only endure extreme cold, but continue metabolic activity, form structured communities, and interact with other organisms to sustain an ecosystem. In this sense, cyanobacteria were more than survivors. They were pioneers, laying the foundation for the future diversification of life. During Snowball Earth, these tiny cells also exhibited remarkable dispersal abilities. Wind-blown ice fragments, meltwater flows, and subglacial currents could carry colonies to new habitats, allowing cyanobacteria to colonize areas that were periodically thawed or enriched with nutrients from volcanic activity or chemical upwelling. Their small size, combined with metabolic flexibility and protective biofilms, made them adept at exploiting transient opportunities, ensuring that populations could persist despite the global ice cover. Perhaps the most inspiring aspect of cyanobacteria survival was their persistence in isolation. Some populations likely survived for thousands of years in extremely limited refuges, such as geothermal vents or brine pockets beneath kilometers of ice. In these isolated niches, cyanobacteria faced extreme scarcity, fluctuating salinity, and near-total darkness, yet they endured. Their ability to survive such conditions underscores the principle that resilience is not solely a matter of size or complexity. Even the simplest life forms can persist against overwhelming odds. Cyanobacteria's story during Snowball Earth is one of ingenuity, endurance, and quiet heroism. By capturing faint light, forming protective mats, engaging in symbiosis, and maintaining metabolic flexibility, they preserved life through one of Earth's most severe glaciations. Without them, the fragile ecosystems beneath the ice would have collapsed, and the chain of life that leads to complex organisms, including humans, may have been broken. These microscopic pioneers remind us that even the tiniest organisms can have an outsized impact on the history of life. Algae, the green survivors of the ice, beneath the thick, unyielding ice of Snowball Earth, where sunlight was a scarce and precious commodity, algae quietly persevered, turning frozen oceans into pockets of life and hope. Unlike cyanobacteria, which were single-celled and extremely resilient, algae represented a higher level of complexity, a eukaryotic lineage capable of photosynthesis, nutrient storage, and adaptive growth in fluctuating environments. During the cryogenian glaciations, algae played a pivotal role, sustaining not only themselves, but entire micro-ecosystems that relied on their energy production and oxygen output. Algae's success in these extreme conditions was driven by an astonishing array of survival strategies. First, many species adopted low-light adaptations that allowed them to capture the faint sunlight penetrating through thick ice sheets or snow-covered surfaces. Pigments such as chlorophyll A and C, as well as accessory pigments like carotenoids and phycobilins, enabled them to absorb wavelengths of light that others could not, maximizing energy capture under near-total darkness. Some species formed dense mats or colonies on the underside of the ice, creating networks of filaments or sheets that both stabilized them against currents and enhanced light absorption. These structures also provided surfaces for other microorganisms to cling to, forming miniature, layered ecosystems beneath the ice. Another critical adaptation was nutrient storage and metabolic flexibility. Algae could store carbon and energy as starch or lipids, enabling them to survive periods of extreme scarcity. In environments where nutrients were limited, such as subglacial brine channels or frozen meltwater pools, these reserves allowed algae to slow growth and maintain cellular integrity. Some species even developed the ability to metabolize alternative energy sources, switching from photosynthesis to consuming organic compounds in dark conditions, ensuring survival when sunlight was unavailable. This metabolic flexibility was crucial for enduring the extended periods of darkness that characterize Snowball Earth winters, or for surviving under meters of ice that blocked most light from penetrating. Algae were not passive survivors. They actively engineered their environment. By forming mats and biofilms, they stabilized sediments and created protective niches for other organisms, including protozoa, bacteria, and even the earliest multicellular animals. These mats trapped organic matter, provided oxygen through photosynthesis, and facilitated nutrient cycling. In doing so, algae became critical ecosystem engineers, transforming marginal habitats into viable refuges where life could persist through the glaciations. Their contribution was subtle yet foundational. Without algae maintaining energy flow and oxygen levels, complex life would have faced even greater hurdles to survival. Symbiosis was another key component of algae's survival strategy. Many algae engaged in partnerships with bacteria, archaea, and even early sponges. These relationships allowed them to acquire essential nutrients, detoxify harmful compounds, or supplement their energy needs. For example, nitrogen-fixing bacteria could convert atmospheric nitrogen into forms usable by algae, compensating for nitrogen scarcity under the ice. Likewise, symbiotic bacteria could remove waste products or enhance photosynthetic efficiency, helping algal communities maintain metabolic balance in extreme conditions. These cooperative strategies highlight a recurring theme of Snowball Earth survival. No organism endured alone. Persistence depended on networks of interactions. Fossil evidence underscores algae's presence and diversity during the cryogenian period. Microscopic impressions, preserved as organic walled microfossils, reveal filamentous and colonial structures remarkably similar to modern algae. These fossils indicate that algae were widespread, thriving in shallow, brine-rich environments. subglacial pools, and even near geothermal vents where chemical energy supplemented limited sunlight. Their distribution suggests that algae were not only surviving, but actively contributing to the structuring of early marine ecosystems, setting the stage for post-glacial biodiversity. Algae also displayed remarkable dispersal abilities. Tiny motile cells could drift with subglacial currents or be transported through meltwater flows, colonizing new niches as ice sheets shifted and fractured. Some formed resistant cysts or spores capable of surviving freezing, nutrient scarcity, and prolonged darkness. These dormant stages allowed populations to persist through extreme environmental fluctuations, reactivating when conditions improved. This strategy ensured that even the harshest periods of Snowball Earth could not eradicate algal populations entirely, providing continuity for subsequent evolutionary developments. The ecological importance of algae extended beyond the immediate survival of their own species. By producing oxygen and organic matter, they supported a variety of heterotrophic organisms, including protists, early animals like sponges, and microscopic crustaceans. Oxygen production, though limited under ice, created pockets of habitable water, gradually altering the chemical environment in ways that would facilitate more complex life. Their role as primary producers was indispensable. In a frozen world where energy sources were scarce, algae were the solar panels of Snowball Earth, capturing every photon and converting it into usable energy for the wider ecosystem. Modern analogues of cryogenian algae demonstrate the enduring nature of these survival strategies. Polar and glacial algae thrive beneath Antarctic and Arctic ice, adapting to extremely low light, sub-zero temperatures, and seasonal nutrient scarcity. Some species even produce pigments that protect against ultraviolet radiation during brief periods of sun exposure, a trait likely shared by their cryogenian ancestors. These modern organisms reflect the evolutionary ingenuity that enabled algae to persist during one of Earth's most extreme glaciations, illustrating that the solutions developed millions of years ago remain effective today. Algae also contributed to the chemical stability of their habitats, influencing the cycles of carbon, nitrogen, and sulfur. By sequestering carbon into biomass, they prevented the accumulation of harmful compounds in confined brine channels. Nitrogen-fixing symbionts ensured a continual supply of essential nutrients, while interactions with sulfur-metabolizing bacteria helped regulate chemical imbalances. These activities highlight algae not just as survivors, but as ecosystem stabilizers, maintaining environmental conditions that allowed other organisms to persist through Snowball Earth's extreme freeze. The story of algae during Snowball Earth is one of resilience, ingenuity, and quiet heroism. They were far more than passive cells. They were ecosystem engineers, energy producers, and stabilizers of chemical and biological balance. By adapting to extreme light scarcity, fluctuating nutrients, and freezing temperatures, algae maintained the flow of energy through ecosystems that otherwise teetered on the edge of collapse. Their survival ensured that, when the ice began to thaw, life had a foundation upon which to rebuild, allowing more complex organisms to thrive in newly accessible environments. Archaea, the chemical survivors of Snowball Earth. Beneath the layers of ice that blanketed the planet during Snowball Earth, where sunlight barely penetrated and photosynthesis was limited, life persisted in forms that thrived on chemistry rather than light. Archaea, single-celled microorganisms, often considered the most ancient and resilient of all life forms, became masters of survival in these extreme conditions. Unlike cyanobacteria or algae, Archaea could exploit chemical energy from their environment, metabolizing substances such as sulfur, methane, hydrogen, and ammonia to sustain life where light could not reach. In brine channels trapped under kilometers of ice, in geothermal vents along the seafloor, and in deep, isolated pockets of seawater, Archaea carved out habitats that were inhospitable to almost all other organisms. Many species were extremophiles, adapted to temperatures, salinities, and chemical concentrations that would be lethal to most life forms. Thermophilic Archaea thrived near hydrothermal vents, where heat and mineral-rich water provided a reliable source of energy. Halophilic species concentrated in brine pockets, tolerating salinity levels far above the open ocean, while acidophilic and alkaliphilic types exploited chemically extreme niches formed by rock-water interactions and mineral leaching under the ice. Archaea were capable of extraordinary metabolic flexibility, switching between energy sources depending on what was available. Methanogens converted carbon dioxide and hydrogen into methane, providing both energy for themselves and, indirectly, for other organisms in the ecosystem. Sulfur-reducing Archaea could metabolize sulfates and sulfides, generating energy in oxygen-poor environments while influencing the chemical composition of their surroundings. This metabolic adaptability allowed Archaea to persist in environments where sunlight-driven photosynthesis was nearly impossible, giving them access to energy sources that were stable and continuous even during prolonged darkness. Many Archaea formed biofilms, similar to cyanobacteria and algae, attaching to rock surfaces, sediments, or microbial mats. These biofilms served multiple purposes. They concentrated nutrients, stabilized cells against flowing meltwater, and allowed cooperative interactions with other microorganisms. In some cases, Archaea lived in symbiosis with bacteria or algae, exchanging metabolites in mutually beneficial arrangements. For example, methane produced by methanogens could be used by other microbes as an energy source, creating miniature chemical cycles that sustained life in an otherwise static environment. Reproduction and dormancy were essential to archaeal survival. Many species could enter a dormant or low metabolic state during periods of extreme stress, such as freezing, desiccation, or nutrient scarcity. Encysted cells, or spores, remained viable for years or decades, reactivating when conditions improved. This capacity for extended dormancy ensured that even drastic changes in environmental conditions, such as thickening ice sheets, shifts in salinity, or temporary depletion of chemical energy, did not eliminate archaeal populations entirely. The presence of Archaea also had profound implications for other organisms beneath the ice. By metabolizing chemical compounds and altering local chemical gradients, they influenced nutrient availability and the stability of microhabitats. Their chemical transformations supported microbial mats, provided energy sources for protists and small metazoans, and created microenvironments suitable for the earliest multicellular life. Archaea acted as the quiet engineers of Snowball Earth ecosystems, sustaining energy flows where photosynthesis alone was insufficient. Modern extremophilic Archaea illustrate how these survival strategies could have functioned during Snowball Earth. Organisms inhabiting hydrothermal vents, deep-sea brine pools, and Antarctic subglacial lakes exhibit remarkable resistance to cold, salinity, and low oxygen, thriving on chemical energy in complete darkness. Their cellular structures are designed to withstand environmental stress with protective membranes, specialized enzymes, and DNA-stabilizing proteins that prevent damage from extreme conditions. These adaptations mirror the capabilities that would have allowed cryogenia and Archaea to persist for millions of years, suggesting a continuity of survival strategies across geological time. Archaea also contributed to the stability of biogeochemical cycles under the ice. Methanogenic and sulfur-metabolizing archaea influenced carbon and sulfur fluxes, preventing toxic accumulation of compounds and ensuring that essential nutrients remained available to other life forms. Their metabolic activity created gradients of chemicals such as hydrogen sulfide, methane, and ammonia, which in turn provided energy for symbiotic microbes, protists, and even early animals like sponges. In this sense, archaea were both consumers and facilitators, supporting broader ecosystems despite the extreme conditions. Interactions between archaea and other microorganisms were dynamic and spatially organized. In brine channels, dense biofilms of archaea coexisted with cyanobacteria, algae, and bacteria, each occupying specific layers optimized for energy capture and nutrient exchange. Photosynthetic organisms utilized available light near ice surfaces, while archaea dominated the darker, deeper layers, converting chemical energy into biomass that supported the food web. Such stratification created pockets of stability in an otherwise hostile environment, allowing multiple forms of life to survive in close proximity. The survival of archaea during Snowball Earth illustrates a broader principle of life under extreme conditions. Flexibility, cooperation, and exploitation of alternative energy sources are critical for persistence. While visible organisms like algae and sponges captured attention by their structure or activity, archaea were the invisible backbone, metabolically sustaining communities and maintaining chemical continuity across frozen oceans. They exemplify the idea that life can endure not just by adapting to one type of energy or niche, but by mastering multiple pathways and integrating into networks of interdependent survival. Archaea's endurance also hints at their evolutionary significance. By thriving in extreme conditions, they preserved lineages that could later diversify and contribute to complex ecosystems. Methanogens, sulfur metabolizers, and other archaeal groups maintained metabolic strategies that would be foundational for future microbial communities, influencing oxygen availability, nutrient cycles, and the chemical balance of the oceans. Their presence ensured that even in the darkest, coldest recesses, life could persist and eventually facilitate the rise of multicellular organisms in the post-glacial world. Protists, the single-celled navigators. Beneath the frozen expanse of Snowball Earth, where ice stretched across oceans and sunlight filtered only weakly through cracks and thin sheets, single-celled protists thrived as some of the most versatile and dynamic survivors. These eukaryotic microorganisms, ranging from amoeboid shapeshifters to flagellated swimmers, were masters of adaptation, navigating the frozen waters with strategies that combined mobility, metabolic flexibility, and ecological intelligence. Unlike multicellular organisms, which relied on structural complexity for survival, protists survived largely through their ability to respond quickly to environmental changes, exploit transient microhabitats, and engage in intricate interactions with other microscopic life. Protists occupied virtually every niche beneath the ice. In brine channels, they floated freely, capturing bacteria, cyanobacteria, and smaller protists as food. Others adhered to the surfaces of microbial mats, moving slowly through layers of sticky biofilm in search of nutrients. Some lived symbiotically within the structures of algae or archaea, exchanging metabolites and forming miniature ecosystems at a cellular level. Their small size allowed them to access crevices and microenvironments inaccessible to larger organisms, exploiting pockets of water that were slightly warmer, more oxygenated, or richer in nutrients than surrounding areas. Amoeboid protists, with their shape-shifting ability, could extend pseudopodia to engulf prey, navigate complex environments, and anchor themselves temporarily when currents threatened to displace them. This ability to alter form in response to environmental cues allowed them to thrive in areas with fluctuating flow, temperature, and nutrient availability. Some species insisted, entering dormant states when conditions worsened, protecting themselves from freezing, starvation, or chemical stress until circumstances improved. In this way, protists combined mobility and dormancy in a balance that allowed them to endure extreme conditions for extended periods. Flagellated protists employed a different strategy, using whip-like appendages to propel themselves through brine channels or meltwater streams. Their motility allowed them to actively seek out food, oxygen, or microhabitats with favorable conditions, rather than passively waiting for nutrients to drift by. In turbulent or unstable environments beneath ice, this mobility was crucial. It enabled protists to relocate when currents shifted, when local nutrient supplies were depleted, or when brine channels became temporarily inhospitable. Photosynthetic protists, including green and red algae-like species, augmented energy flow in these frozen ecosystems. Even under dim light, their photosynthetic pigments captured faint sunlight filtered through the ice, producing organic matter that fueled local food webs. In areas where light was insufficient, some protists adopted mixotrophy, combining photosynthesis with ingestion of bacteria or other protists. This dual strategy allowed them to maintain energy balance and sustain growth even when environmental conditions fluctuated unpredictably, a common feature beneath the ice. Protists also played key ecological roles, shaping the structure and dynamics of Snowball Earth ecosystems. By consuming bacteria, cyanobacteria, and smaller protists, they regulated population densities, preventing any single species from dominating and depleting local resources. Their movement through sediments and microbial mats redistributed nutrients, facilitating the growth of photosynthetic organisms and supporting higher trophic levels, including early metazoans like sponges or small multicellular invertebrates. In effect, protists were the traffic controllers of microscopic ecosystems, orchestrating energy flow and nutrient cycling in a frozen, sparsely populated world. Symbiotic relationships further enhanced protist survival. Some protists hosted photosynthetic symbionts, such as cyanobacteria or algae, allowing them to harness chemical energy from photosynthesis while remaining mobile and heterotrophic. Others engaged in mutualistic interactions with archaea or bacteria, exchanging waste products for nutrients or chemical energy. These partnerships created microhabitats of heightened stability and resource availability, increasing the likelihood of persistence across long glacial periods. Reproduction among protists combined rapid proliferation with survival strategies. Asexual reproduction allowed populations to expand quickly when conditions were favorable, while sexual processes, or the formation of cysts, preserved genetic diversity and resilience. Resistant cysts could endure freezing, darkness, or nutrient scarcity for years, reactivating when ice thinned or meltwater delivered fresh nutrients. This dual reproductive strategy ensured continuity of populations even during prolonged periods of environmental stress, allowing protists to maintain a constant presence in the frozen oceans. Fossil evidence, though limited due to the microscopic and delicate nature of protist bodies, suggests that these organisms were widespread during the Cryogenian. Organic walled microfossils, sometimes preserved in association with microbial mats, reveal filamentous, amoeboid, and flagellated forms that closely resemble modern protists inhabiting polar and glacial environments. These findings indicate that protists were not only surviving but actively shaping the microecological dynamics of Snowball Earth, influencing nutrient cycling, oxygen availability, and the distribution of other microorganisms. Modern analogues of Snowball Earth protists provide a window into their ancient adaptations. Antarctic and Arctic lakes, subglacial meltwater streams, and deep-sea environments host protists that endure near-freezing temperatures, extreme salinity, and prolonged darkness. Many exhibit behaviors directly comparable to cryogenian forms: amoeboid movement, cyst formation, mixotrophy, and symbiosis. These contemporary organisms highlight the evolutionary continuity of survival strategies that allowed protists to navigate some of the most extreme conditions in Earth's history. Protists were also critical intermediates between microbial life and early multicellular organisms. By forming concentrated colonies, interacting with algae, cyanobacteria, and archaea, and facilitating nutrient cycling, they created microhabitats capable of supporting more complex life. Their presence enhanced the stability of microbial mats, influenced oxygen and carbon availability, and contributed to the chemical and physical structuring of the benthic environment. These contributions were subtle but essential, ensuring that when Snowball Earth temporarily thawed in localized areas, life could expand rapidly into newly accessible niches. The mobility, metabolic versatility, and ecological integration of protists allowed them to occupy a wide range of habitats beneath the ice. They were found in shallow brine channels where currents delivered nutrients, in subglacial pools that remained liquid due to geothermal heat, and even in sediment layers beneath microbial mats, feeding on detritus and bacteria. By colonizing such diverse microenvironments, protists maintained continuity of life, acting as a bridge between simple microorganisms like cyanobacteria and algae, and the earliest multicellular animals that would later populate the oceans. In addition to sustaining life beneath the ice, protists contributed to long-term evolutionary processes. By preserving genetic diversity, experimenting with metabolic flexibility, and maintaining ecological networks, they set the stage for the eventual emergence of larger, more complex organisms. Their interactions with other microbial and eukaryotic species fostered selective pressures that drove adaptation, ultimately influencing the trajectory of life during and after the cryogenian glaciations. Protists were the navigators of Snowball Earth, exploring the frozen oceans with unmatched versatility. They moved, fed, reproduced, and adapted in a world where every resource was scarce and every habitat was precarious. Their ability to integrate into microecosystems, exploit chemical and photosynthetic energy sources, and endure extreme conditions ensured that life remained dynamic and interconnected despite the icy isolation imposed by global glaciation. Sponges, the earliest multicellular animals. In the frozen oceans of Snowball Earth, where microbial life dominated and sunlight barely reached beneath thick ice sheets, some of the earliest multicellular animals carved out a quiet but persistent existence. Sponges, simple yet remarkably resilient, represent the first known animals capable of sustaining themselves in a world dominated by extreme cold, scarcity of nutrients, and highly variable environmental conditions. Their structural simplicity, combined with intricate cellular specialization, allowed them to survive where other multicellular organisms might have perished. Sponges, or porophorans, lack true tissues, organs, or nervous systems, yet their bodies are extraordinarily efficient at performing the fundamental tasks of life. Their internal canals and chambers, lined with specialized cells called choanocytes, generate water flow, capture microscopic food particles, and facilitate gas exchange. This passive, yet highly effective system allowed sponges to extract nutrients from water that flowed over or through them, even in nutrient-poor environments beneath kilometers of ice. Their sessile lifestyle, anchored to rocky substrates or microbial mats, further increased survival by allowing them to remain in stable microhabitats where currents delivered a steady supply of bacteria, detritus, and other microscopic food sources. The survival of sponges during Snowball Earth relied heavily on their cellular flexibility. Different cell types within their bodies could adapt to changes in nutrient availability, water flow, and temperature. Choanocytes, responsible for capturing food, could regulate water pumping rates to optimize energy intake, while archaeocytes moved nutrients internally, distributing resources to support growth or maintenance. Some cells even entered semi-dormant states during extreme scarcity, conserving energy until conditions improved. This division of labor, combined with modular body organization, allowed sponges to endure environments that would challenge more complex multicellular life. Sponges were also highly adaptable in their reproductive strategies. Many produced resistant larvae capable of surviving harsh conditions, including freezing temperatures and low nutrient availability. These larvae could drift with currents until they found suitable attachment points, ensuring the continuity of populations across the frozen oceans. Some sponges may have reproduced asexually through budding or fragmentation, allowing them to colonize local habitats rapidly and maintain genetic continuity even when sexual reproduction was limited by environmental stress. Their feeding strategies provided another key to survival. Sponges are filter feeders, able to process thousands of liters of water relative to their size each day. This capability allowed them to extract the sparse bacterial and protist biomass available in sub-ice waters efficiently. In addition, some sponges may have relied on symbiotic relationships with bacteria and cyanobacteria, supplementing their nutrition with energy produced through photosynthesis or chemical metabolism. These partnerships created microhabitats where energy and nutrients were recycled efficiently, stabilizing local ecosystems and enhancing the persistence of both sponges and their microbial partners. Fossil evidence suggests that sponges were already present during the cryogenian glaciations, though their delicate bodies left behind only sparse traces. Microscopic spicules, made of silica or calcium carbonate, have been discovered in sedimentary deposits dating back to this period. These structures not only confirm their presence, but also indicate that sponges were sufficiently abundant to contribute to sediment stabilization and benthic community structure. By anchoring themselves and filtering water, sponges played an ecological role disproportionate to their size, influencing nutrient distribution and providing surfaces for microbial colonization. Sponges also demonstrated remarkable resilience to environmental stress. Their porous bodies allowed them to tolerate variations in water chemistry, salinity, and temperature, common in brine channels and meltwater pockets beneath the ice. The modular nature of their structure meant that damage to one part of the body did not necessarily compromise overall survival. Even partial fragmentation could lead to the development of new individuals, a strategy that proved advantageous in environments where ice movement, currents, or sediment shifts could physically disrupt organisms. Modern analogues of Snowball Earth sponges provide insight into their ancient strategies. Species inhabiting polar regions today survive near freezing temperatures, low light, and seasonal scarcity of nutrients, often relying on symbiotic microorganisms to supplement their diet. These contemporary sponges exhibit the same modular growth patterns, filter feeding, efficiency, and resilience that would have been essential for survival during the cryogenian glaciations. Their continued existence demonstrates the evolutionary success of the adaptations developed millions of years ago in response to extreme environmental conditions. Sponges were also pivotal in shaping early benthic ecosystems. By filtering water, they facilitated nutrient cycling and oxygen distribution, indirectly supporting the survival of other microorganisms, such as protists, bacteria, and algae. Their physical presence created structural complexity, providing surfaces for microbial mats and small motile organisms to cling to or navigate. In this way, sponges acted as ecosystem engineers, contributing to the spatial organization and stability of early multicellular communities beneath the ice. Mobility was not a feature of sponges themselves, but their larvae contributed to dispersal and colonization. Free-swimming larvae could navigate currents and settle in microhabitats with favorable conditions, ensuring genetic flow and population maintenance. This combination of sessile adults and mobile larvae provided a balance between stability and exploration, enabling sponges to persist across a fragmented, frozen world where habitable zones were patchy and isolated. The cryogenian oceans, dominated by ice and brine, were harsh and unforgiving, yet sponges thrived by optimizing simplicity, flexibility, and cooperation. Their bodies functioned as living filtration systems, their cells as specialized tools for survival, and their partnerships with microorganisms as mutualistic networks that maximized energy capture and nutrient recycling. Sponges exemplified how multicellular life could endure extreme conditions without complexity or speed, relying instead on efficiency, modularity, and integration with surrounding ecosystems. Sponges' presence beneath the ice also set the stage for the diversification of more complex animals after Snowball Earth. By maintaining benthic microhabitats, stabilizing sediments, and facilitating energy flow through microbial partnerships, they provided the ecological scaffolding that allowed early multicellular animals to thrive in post-glacial oceans. Their survival strategies reflect a principle that recurs throughout Earth's history. Life can persist in extreme conditions not by resisting change, but by integrating with its environment and exploiting every opportunity for energy, stability, and continuity. Early arthropods, microscopic pioneers. Beneath the frozen, near-lightless oceans of Snowball Earth, life was not limited to microbial mats and microscopic single-celled organisms. Some of the earliest ancestors of arthropods, the highly successful group that would later dominate Earth's oceans and land, found ways to persist in this extreme environment. These tiny, often microscopic creatures relied on a combination of mobility, protective exoskeletons, and highly adaptable feeding strategies to survive in oceans trapped under thick sheets of ice. Navigating a world where energy was scarce and environmental conditions shifted rapidly, early arthropods were among the first animals to exploit active locomotion beneath the ice. Unlike sessile sponges, they could swim or crawl through brine channels, exploring microhabitats and pursuing microscopic prey. Their segmented bodies and jointed appendages provided flexibility and control, allowing them to move efficiently in turbulent meltwater flows or navigate narrow gaps between ice crystals and microbial mats. Mobility was a key advantage, enabling them to access nutrient-rich pockets, avoid predators, and colonize areas that were inhospitable to slower or stationary organisms. Protective adaptations were central to arthropod survival. Even at a microscopic scale, early arthropods developed exoskeletons composed of chitin or similar substances, providing structural support, defense against environmental hazards, and protection from microbial overgrowth. In the freezing waters of Snowball Earth, these exoskeletons offered insulation, reducing heat loss and helping maintain cellular integrity during extreme cold. They also served as barriers against physical stress from shifting ice, sediment, and brine currents, ensuring that even small creatures could persist in unstable habitats. Feeding strategies among early arthropods were diverse and highly specialized. Some were filter feeders, passing water through appendages adapted to trap bacteria, protists, and algae. Others were grazers or scavengers, scraping microbial mats or consuming detritus deposited on the ocean floor. Predatory forms likely hunted smaller protists, ciliates, and juvenile arthropods, creating the earliest microfood webs beneath the ice. This diversity of feeding modes reduced competition, allowing multiple arthropod species to coexist in the same environment while exploiting different resources. Reproduction was a critical component of persistence under extreme conditions. Many early arthropods produced resistant eggs capable of surviving freezing, low oxygen, and nutrient scarcity. These eggs could be dispersed by currents, settling in suitable microhabitats when environmental conditions improved. Rapid asexual reproduction may have allowed populations to expand quickly in localized refuges, while sexual reproduction maintained genetic diversity, facilitating long-term adaptation. This combination of reproductive strategies ensured both short-term survival and evolutionary continuity. Early arthropods also engaged in complex behavioral adaptations. Their mobility allowed them to track shifting resource availability, respond to chemical cues, and form temporary aggregations in favorable microhabitats. Some species may have sought shelter beneath microbial mats or within brine channels, exploiting structural complexity to avoid harsh currents or predation. This capacity for behavioral response was an important survival strategy, enabling them to exploit transient niches and maintain populations in a world of extreme environmental flux. Interactions with other organisms were integral to survival. Early arthropods coexisted with protists, cyanobacteria, algae, archaea, and sponges, forming the first structured food webs in frozen oceans. By grazing on microbial mats or consuming protists, they controlled population densities and facilitated nutrient cycling. Some may have carried symbiotic microorganisms on their surfaces or within specialized tissues, creating microecosystems that enhance nutrient absorption or chemical stability. In these ways, early arthropods were both consumers and facilitators, shaping the microscopic ecosystems that persisted beneath the ice. The fossil record provides glimpses of these early arthropods, though their tiny and delicate forms left sparse traces. Exoskeleton fragments, microscopic claws, and evidence of segmented bodies have been discovered in cryogenian sediments, indicating that arthropods were already experimenting with mobility, predation, and environmental specialization. These findings suggest that even at this early stage, arthropods were capable of navigating complex and challenging habitats, exploiting ecological niches that few other multicellular organisms could occupy. Modern analogues offer insight into the survival strategies of these microscopic pioneers. Polar copepods, water fleas, and other small crustaceans endure extreme cold, low oxygen, and limited food availability in contemporary glacial and sub-ice ecosystems. Their ability to enter dormant states, rely on stored energy reserves, and exploit microhabitats mirrors the adaptations early arthropods likely employed. By studying these living descendants, scientists gain a clearer understanding of how mobility, protective structures, and feeding versatility allowed tiny animals to persist in Snowball Earth's frozen oceans. Early arthropods' contribution to ecosystem stability extended beyond direct feeding. By moving through water and sediments, they redistributed organic material, promoting decomposition, nutrient cycling, and the growth of microbial communities. Their presence increased habitat heterogeneity, providing opportunities for protists, sponges, and algae to thrive. These interactions highlight the importance of small, mobile animals in maintaining energy flow and ecological balance, even in a world dominated by ice and microscopic life. The evolution of jointed appendages, sensory structures, and motile behavior in early arthropods provided a foundation for the remarkable diversification that would follow after the glaciations. By exploring microhabitats, exploiting food sources, and surviving extreme conditions, they demonstrated the adaptive potential of multicellular life. Their success beneath Snowball Earth illustrates the principle that mobility, structural innovation, and ecological integration can enable organisms to thrive where more stationary or less versatile life forms might fail. Early arthropods were the silent navigators of frozen oceans, moving through brine channels, microbial mats, and sediment layers, capturing energy and distributing nutrients across the ecosystem. Their adaptations ensured that life remained interconnected and dynamic, setting the stage for larger, more complex animals to eventually populate the post-glacial oceans. Their survival strategies, mobility, protective exoskeletons, diverse feeding modes, and reproductive resilience exemplify the ingenuity of life in one of Earth's most extreme chapters. Sea cnidarians, early predators and reef builders. In the dimly lit oceans beneath the global ice of Snowball Earth, life had begun experimenting with new strategies of survival and predation. Among these early multicellular organisms, cnidarians, ancestors of modern jellyfish, corals, and sea anemones, found ways to persist. Simple in organization, yet remarkably effective, cnidarians combined basic tissue-level specialization with specialized feeding structures to capture prey, navigate water currents, and interact with other organisms. Their presence in the cryogenian seas illustrates how early animals were not only surviving, but beginning to structure ecological communities beneath the ice. Cnidarians were among the first true predators. Equipped with specialized stinging cells called cnidocytes, they could immobilize microscopic prey, such as protists, bacteria, and small planktonic arthropods. These cells contain tiny capsules filled with toxins and harpoon-like structures, enabling cnidarians to deliver precise, rapid stings. In a frozen world where energy was scarce and opportunities for feeding were rare, this adaptation provided a critical advantage, allowing them to capture food more efficiently than passive filter feeders. Tentacles lined with cnidocytes formed feeding arrays that could be extended into currents or retracted when danger loomed, offering both mobility and protection in the microhabitats beneath the ice. Despite their predatory capabilities, many cnidarians were sessile, anchored to rock surfaces, microbial mats, or early reef structures formed by microbial and algal mats. This sessile lifestyle allowed them to stabilize in nutrient-rich microenvironments while extending tentacles to capture passing prey. Others developed a more free-floating medusa form, drifting through brine channels and open water, capturing microorganisms and exploiting sparse energy sources wherever currents carried them. This duality, sessile, polyps, and mobile medusae, enabled cnidarians to occupy multiple ecological niches simultaneously, enhancing their chances of survival during extended periods of ice cover. Reproduction in cnidarians was versatile, supporting survival under extreme conditions. Many species reproduced asexually through budding or fragmentation, enabling them to expand rapidly in localized refuges or recover from environmental damage. Sexual reproduction produced resistant larvae, capable of drifting with currents until suitable microhabitats were located. These larvae could endure low temperatures, darkness, and nutrient scarcity, emerging in optimal conditions to establish new populations. The combination of asexual expansion and sexually dispersed larvae ensured genetic continuity and increased the resilience of cnidarian populations throughout the Cryogenian. Cnidarians also relied on symbiotic relationships to augment their energy intake. Some species harbored photosynthetic symbionts, such as cyanobacteria or microalgae, allowing them to supplement their nutrition with chemical energy captured from even the faintest sunlight filtering through the ice. In areas where light was insufficient, cnidarians maintained heterotrophic feeding, capturing prey to meet their metabolic needs. This combination of autotrophic and heterotrophic strategies, known as mixotrophy, provided flexibility in a world where environmental conditions fluctuated unpredictably and energy sources were patchily distributed. The role of cnidarians extended beyond individual survival. They were important ecosystem engineers. Sessile polyps, anchored to substrate-stabilized sediments, created structure for microbial mats to attach to, and facilitated the establishment of small, spatially organized communities. Even tiny, isolated cnidarian colonies could influence local water flow, nutrient distribution, and prey abundance, shaping the microhabitats in which other organisms, including early sponges, protists, and microscopic arthropods, thrived. Their predatory behavior also maintained balance among microbial populations, preventing any single group from overwhelming limited food resources. Fossil evidence from the Cryogenian suggests that cnidarians were already experimenting with both polyp and medusa forms. Early impressions, preserved in sedimentary deposits, indicate radial symmetry, tentacle arrangements, and body plans consistent with later cnidarian lineages. These findings imply that the basic blueprint of cnidarian organization tissues, cnidocytes, and radial symmetry was already established, equipping them for survival under extreme conditions. Even in the absence of abundant light and with limited energy resources, cnidarians were successfully feeding, reproducing, and interacting with other organisms, forming the earliest structured animal communities. Modern analogues illustrate how these ancient survival strategies could have functioned. Polar jellyfish, subglacial sea anemones, and deep-sea cnidarians today endure near-freezing temperatures, low oxygen levels, and extended periods of darkness, while maintaining predatory and reproductive capabilities. Some host photosynthetic symbionts that supplement nutrition, a strategy likely shared with Cryogenian counterparts. Observations of these contemporary organisms offer insight into how early cnidarians navigated a frozen, energy-limited world, balancing predation, symbiosis, and mobility to persist through extreme conditions. Cnidarians' impact on Snowball Earth ecosystems extended to micro-scale structural complexity. By forming small colonies, they provided surfaces for protists and algae to attach, created vertical space in otherwise uniform environments, and generated localized gradients of prey abundance and oxygen levels. Their predatory activity redistributed microbial populations, influencing nutrient cycling and energy flow. Even at small sizes, cnidarians introduced dynamics that shaped the interactions of multiple species, fostering an interconnected web of life beneath the ice. The evolutionary significance of cnidarians during Snowball Earth cannot be understated. Their ability to combine structural simplicity with functional specialization, predation, symbiosis, and modular reproduction demonstrated the potential of multicellular life to adapt to extreme environments. These strategies laid the foundation for the diversity of cnidarian lineages that would later dominate reef systems, pelagic zones, and benthic communities after the glaciations. By maintaining micro-ecosystems, capturing scarce energy, and facilitating ecological interactions, cnidarians contributed to the continuity and diversification of life in ways that extended far beyond their immediate survival. Cnidarians were both predators and architects, capable of exploiting prey while shaping habitats for themselves and others. They integrated with microbial mats, anchored to substrates, drifted through currents, and sustained energy flow in frozen oceans where sunlight barely penetrated. Through mobility, predation, symbiosis, and reproductive resilience, they exemplified the versatility of early animals and the ability of multicellular life to persist in extreme conditions. Their presence beneath Snowball Earth illustrates how simple body plans, combined with functional ingenuity, allowed life to maintain complexity and continuity, even when global environments approached near total inhospitability. Early mollusks, the first shell builders and grazers, amid the frozen waters of Snowball Earth, a new group of multicellular organisms was beginning to make its mark beneath the ice. Early mollusks. Simple in body plan, yet highly adaptable, these creatures combined protective shells, muscular foot structures, and diverse feeding strategies to survive in an environment where light was scarce, nutrients were limited, and predation pressures were emerging. Their adaptations allowed them to exploit both the benthic surfaces and the water column, interacting with microbial mats, algae, protists, and other small animals to carve out ecological niches that would form the foundation for later molluscan evolution. One of the most critical adaptations of early mollusks was the development of mineralized shells. Even simple, small shells provided a physical barrier against environmental stresses, including freezing temperatures, abrasive ice movements, and predation by microscopic arthropods or cnidarians. Composed primarily of calcium carbonate, these shells' stabilized body structure allowed mollusks to maintain internal fluid balance and protected delicate tissues. In many cases, shells were thin and lightweight, optimized for energy efficiency, allowing these animals to allocate resources to growth, reproduction, and mobility rather than just defense. The muscular foot of mollusks provided a key advantage in navigating the frozen sea floor. By extending, contracting, and adhering to substrates, mollusks could move across microbial mats, rocky surfaces, and sediment layers, accessing food and avoiding unfavorable conditions. This mobility distinguished them from sessile organisms like sponges and polyps, giving mollusks the ability to explore, graze, and escape microhabitats where resources were depleted or predators were present. Their foot also facilitated burrowing, which created additional shelter and allowed them to exploit detritus and microorganisms buried in sediments beneath the ice. Feeding strategies among early mollusks were highly adaptable. Many grazed on microbial mats, scraping surfaces with a primitive radula, a toothed, tongue-like structure used to rasp bacteria, algae, and detritus. Others were filter feeders, drawing water over specialized gills to capture suspended microorganisms. This diversity in feeding modes reduced competition and allowed multiple species to coexist, occupying both benthic and sub-ice water column niches. Some may have combined strategies, grazing when microbial mats were abundant, and filter feeding when planktonic food sources were present, maximizing survival in an environment where energy availability was highly variable. Reproduction in early mollusks supported persistence under extreme cryogenian conditions. Many species produced resistant eggs capable of withstanding freezing, darkness, and nutrient scarcity, while larvae could drift with currents to colonize new habitats. Asexual reproduction, or fragmentation, may have supplemented these strategies in certain species, enabling rapid local population expansion in favorable microhabitats. This combination of reproductive modes enhanced both short-term survival and long-term evolutionary continuity, ensuring that mollusks remained present across the fragmented and dynamic frozen oceans. Mollusks also engaged in symbiotic relationships that enhanced survival. Some hosted photosynthetic microorganisms in their tissues or shells, supplementing nutrition with energy captured from faint sunlight penetrating thin ice. Others relied on associations with bacteria capable of metabolizing chemicals, such as sulfur or methane, providing additional energy sources when photosynthetic inputs were insufficient. These partnerships allowed mollusks to persist in niches where independent feeding would have been inadequate, integrating them into complex microbial and protist-driven ecosystems. The fossil record of early mollusks during the Cryogenian is limited but informative. Tiny shell fragments, preserved in sedimentary layers, indicate that mollusks were experimenting with mineralized protection, mobility, and grazing behavior. These fossils suggest that even at this early stage, mollusks were exploiting benthic habitats, interacting with microbial mats, and contributing to nutrient cycling. Their shells, even when microscopic, stabilized sediments and provided surfaces for microbial colonization, enhancing the structural complexity of the benthic environment and supporting the persistence of other organisms. Modern analogues of cryogenian mollusks provide further insight into their survival strategies. Polar and glacial mollusks, including small bivalves and gastropods, endure near-freezing temperatures, low oxygen levels, and seasonal nutrient scarcity, while using shells, muscular feet, and specialized feeding strategies to persist. Many display behavioral flexibility, moving between grazing on microbial surfaces and filter feeding suspended particles, mirroring the versatility likely employed by their cryogenian ancestors. Observations of these living mollusks demonstrate how early shell builders navigated frozen oceans and exploited diverse energy sources to maintain life in extreme conditions. Early mollusks also contributed to ecosystem structuring. By grazing microbial mats, they regulated bacterial and algal densities, preventing overgrowth and promoting nutrient cycling. Their movement redistributed organic material, creating localized enrichment zones and supporting protists, early arthropods, and cnidarians. Burrowing behavior aerated sediments, facilitating microbial decomposition and stabilizing benthic microhabitats. Even as small, simple animals, mollusks influenced energy flow, habitat heterogeneity, and community dynamics, enhancing resilience and continuity of life beneath the ice. The evolution of shells, motility, and diverse feeding strategies in early mollusks illustrates a principle central to Snowball Earth survival. Structural innovation, combined with ecological flexibility, allows organisms to persist in extreme and unpredictable conditions. By exploiting multiple niches simultaneously, maintaining symbiotic relationships, and reproducing both asexually and sexually, mollusks ensured that they remained integral players in the cryogenian oceans. Their adaptations demonstrate how relatively simple, multicellular animals could persist, diversify, and interact with the broader ecosystem, setting the stage for more complex forms of life to emerge after the glaciations. Mollusks' presence beneath the ice highlights the growing complexity of Snowball Earth ecosystems. They were grazers, filter feeders, burrowers, and symbiont hosts, integrating mobility, protection, and ecological function into a single organism. By stabilizing sediments, redistributing nutrients, and interacting with microbial mats, protists, and other multicellular organisms, mollusks contributed to the foundation of benthic food webs. Their adaptations allowed them to endure extreme cold, scarce resources, and environmental instability, illustrating the combination of flexibility, structural innovation, and ecological integration that characterizes life's persistence in one of Earth's most challenging periods. Early echinoderms, the spiny pioneers of the frozen oceans. In the frozen, dimly lit oceans of Snowball Earth, another remarkable group of multicellular organisms began to establish themselves. Early echinoderms. These spiny, radially symmetrical animals, ancestors of modern starfish, sea urchins, and crinoids, exhibited unique structural and functional adaptations that allowed them to survive in an environment dominated by ice, cold, and limited nutrient availability. Their presence beneath the ice demonstrates how the first animals began experimenting with new body plans, feeding strategies, and ecological roles in an extreme world. One of the key adaptations of early echinoderms was their radial symmetry, which provided structural efficiency and resilience in harsh environments. Unlike bilaterally symmetrical animals, radial symmetry allowed echinoderms to interact with their surroundings in all directions, capturing food, sensing stimuli, and anchoring to substrates without the need for directional movement. This body plan was particularly advantageous in frozen oceans, where currents, ice shifts, and turbulent meltwater could come from any angle. Their symmetrical arrangement of spiny projections or podia facilitated feeding, movement, and defense in a three-dimensional, dynamic environment. Echinoderms were among the first animals to develop a water vascular system, a network of fluid-filled canals that powered movement and feeding. Tube feet extending from these canals allowed echinoderms to grip substrates, navigate uneven surfaces, and manipulate food particles. In the sub-ice environment, where microbial mats, protists, and early mollusks formed dense benthic layers, tube feet provided both mobility and stability. They could crawl across sediment, anchor to rocky surfaces, or extend podia to capture detritus and small microorganisms. This combination of locomotion and feeding efficiency gave echinoderms a distinct advantage in microhabitats with variable nutrient availability and shifting currents. Feeding strategies in early echinoderms were highly diverse. Some grazed on microbial mats, scraping surfaces with spiny structures or podia to access bacteria and algae. Others scavenged detritus, filtering organic particles from sediment or water, while certain forms may have been opportunistic predators, capturing tiny protists or juvenile arthropods. This diversity in feeding modes allowed echinoderms to occupy multiple ecological niches simultaneously, reducing competition and supporting ecosystem complexity. The ability to exploit both benthic and suspended resources was critical in a world where energy inputs were sparse and patchily distributed. Reproduction in early echinoderms contributed to their resilience. Many produced eggs and larvae capable of surviving extreme cold, low light, and nutrient scarcity, dispersing through currents to colonize new habitats. Some species may have employed asexual reproduction through fragmentation, regenerating lost body parts and allowing localized populations to expand without relying on sexual reproduction. The combination of dispersal, resistant larvae, and regenerative capabilities ensured that echinoderms could maintain population continuity even under the environmental stress of global glaciation. Protective adaptations were another key feature. Early echinoderms developed spiny exoskeletons or calcareous plates, offering physical defense against predation and environmental hazards. These structures also provided stability in turbulent subice currents and insulated delicate tissues from cold and chemical stress. Spines and plates may have served additional functions, including anchoring to substrates, manipulating food, or supporting microhabitats for symbiotic microorganisms. By integrating defense, structural integrity, and ecological interaction, early echinoderms optimized survival in a highly challenging environment. Echinoderms also engaged in symbiotic relationships. Their surfaces and tube feet provided habitats for bacteria, protists, and microalgae, which in turn contributed to nutrient cycling and chemical balance. Some echinoderms may have relied on these symbionts for supplemental energy, particularly in areas where food sources were scarce or inconsistent. These micro-ecosystems enhance the overall stability of the benthic environment, supporting diverse communities of microorganisms, protists, mollusks, and other multicellular animals. Fossil evidence indicates that echinoderms were present during the cryogenian glaciations, although preservation is often fragmentary due to the delicate nature of their skeletons. Microscopic plates, spines, and impressions of radial symmetry reveal that these early echinoderms were already experimenting with structural and functional adaptations, including mobility, feeding diversity, and defensive armature. Even at small sizes, echinoderms contributed to sediment stabilization, nutrient cycling, and habitat complexity, playing a role disproportionate to their abundance. Modern analogues offer insights into their survival strategies. Polar sea urchins, brittle stars, and crinoids today endure near freezing temperatures, low oxygen levels, and limited food availability while utilizing tube feet, spines, and radial symmetry to navigate complex microhabitats. Their regenerative abilities, diverse feeding strategies, and symbiotic interactions illustrate how early echinoderms could thrive in cryogenian oceans. Observing these living descendants demonstrates the persistence of evolutionary strategies optimized for survival in extreme environments, including modularity, flexibility, and ecological integration. Echinoderms' interactions with other organisms shaped microecosystems beneath the ice. By grazing microbial mats, consuming detritus, and interacting with protists and small arthropods, they regulated population densities and redistributed nutrients. Their movement aerated sediments and facilitated microbial decomposition, while their structural presence provided surfaces for colonization and microhabitat complexity. In this way, echinoderms acted as both consumers and engineers, enhancing ecosystem resilience and promoting interdependent networks of life beneath the frozen ocean surface. The combination of radial symmetry, spiny protection, water vascular locomotion, and diverse feeding strategies illustrates the adaptive ingenuity of early echinoderms. These traits allowed them to endure environmental extremes, exploit available resources efficiently, and interact with other organisms in ways that stabilized and diversified benthic ecosystems. Their persistence during Snowball Earth demonstrates how relatively simple structural innovations, integrated with behavioral and ecological versatility, can enable multicellular organisms to thrive under conditions that might otherwise seem inhospitable. Early echinoderms, the spiny pioneers of the frozen oceans, exemplified resilience, functional innovation, and ecological integration. They moved through sediment, grazed microbial mats, scavenged detritus, and interacted with microorganisms and other multicellular animals. Through their feeding, locomotion, and symbiotic associations, they contributed to the continuity, complexity, and organization of Snowball Earth ecosystems, ensuring that life beneath the ice remained dynamic, interconnected, and capable of surviving one of Earth's most extreme climatic episodes. Early worms, the burrowing engineers of Snowball Earth. Beneath the global ice sheets of Snowball Earth, where microbial mats, protists, and early multicellular animals formed the foundation of life, early worms, primitive annelids, and related soft-bodied invertebrates emerged as some of the most effective ecosystem engineers. Despite their simple appearance, these organisms played crucial roles in shaping sediments, cycling nutrients, and facilitating the persistence of life in one of the most extreme environments Earth has ever experienced. Their burrowing, feeding, and metabolic activities allowed them to survive in cold, low-light oceans where energy was limited and habitats were spatially fragmented. One of the primary adaptations of early worms was their elongated, flexible body, which allowed them to navigate narrow spaces within sediments, microbial mats, and beneath ice-covered substrates. This mobility enabled worms to exploit microhabitats inaccessible to larger or more rigid organisms. By burrowing through sediment layers, they could access trapped organic matter, bacteria, and detritus, effectively harvesting energy that would otherwise remain unavailable. Their ability to move through multiple layers of sediment increased their survival probability, allowing them to find nutrients, avoid predators, and occupy regions of relative stability within the dynamic, ice-dominated environment. Early worms were remarkable for their sediment engineering capabilities. As they burrowed and consumed organic material, they aerated the sediment, redistributed nutrients, and promoted microbial decomposition. This biotubation enhanced sediment stability, prevented anoxic zones, and created microenvironments favorable for protists, sponges, and other small animals. Even at microscopic scales, the cumulative effects of worm activity transformed the benthic ecosystem, establishing feedback loops that facilitated energy flow and maintained the persistence of life beneath the ice. Feeding strategies among early worms were highly versatile. Many were detritivores, consuming decomposing organic material trapped in sediments or deposited on microbial mats. Others were deposit feeders, ingesting sediment particles and extracting bacterial, protist, and algal nutrients as they passed through the digestive tract. Some species may have been opportunistic predators, preying on smaller invertebrates, protists, or larval forms of arthropods and cnidarians. This diversity in feeding behavior reduced competition and allowed worms to occupy a wide range of ecological niches, making them indispensable components of the Snowball Earth benthic community. The digestive efficiency of early worms played a crucial role in their survival. Specialized gut structures, lined with enzymes and sometimes harboring symbiotic bacteria, allowed worms to extract maximum nutrition from low-energy food sources. Symbiotic microbes assisted in breaking down complex organic compounds, including cellulose or refractory detritus, that would otherwise be inaccessible. These partnerships not only enhanced the worm's energy intake, but also contributed to nutrient cycling in benthic microhabitats, supporting the broader ecosystem and enabling other organisms to persist in an environment of limited resources. Reproductive strategies ensured the resilience and continuity of worm populations. Many early worms reproduced sexually, producing eggs capable of enduring freezing, low oxygen, and periods of starvation. Larvae could disperse through water or sediment, colonizing new microhabitats that became available due to shifts in currents, ice melting, or microbial mat expansion. Asexual reproduction or fragmentation may have supplemented sexual reproduction, enabling worms to maintain or expand local populations even in highly fragmented and extreme environments. This combination of reproductive strategies increased genetic diversity and enhanced survival across the spatially isolated refuges of Snowball Earth. Early worms also exhibited remarkable physiological resilience. Their soft, flexible bodies allowed them to tolerate variations in temperature, salinity, and oxygen levels common in brine channels, subglacial pools, and sediment pockets. The ability to slow metabolism during periods of scarcity, enter dormancy, or reduce activity in response to environmental stress ensured that worms could survive prolonged intervals of resource limitation. These physiological strategies, combined with burrowing and feeding versatility, provided the foundation for their persistence across the most inhospitable habitats beneath the ice. Fossil evidence of cryogenian worms, while rare due to the soft-bodied nature of the organisms, suggests that they were already significant ecosystem participants. Trace fossils, including burrows, tunnels, and feeding marks in sediments, reveal the presence of active burrowers modifying benthic substrates. These patterns indicate that worms were shaping the microenvironment, redistributing organic matter, and creating habitats for smaller organisms. Even in the absence of preserved body structures, the impacts of worms on sediment and nutrient dynamics are evident, demonstrating their critical ecological role. Modern analogues in polar and glacial ecosystems provide insights into the survival strategies of these early worms. Contemporary polychaetes, oligochaetes, and other burrowing invertebrates endure near-freezing temperatures, low oxygen, and limited nutrient availability while manipulating sediment and consuming detritus efficiently. Many rely on symbiotic bacteria to supplement energy intake, reflecting a strategy likely shared by cryogenian worms. Observations of these living species illustrate the persistence of ancient adaptations, including burrowing behavior, digestive versatility, and physiological resilience, all optimized for survival in extreme environments. The ecological contributions of early worms extended beyond their individual survival. By modifying sediment structure, redistributing nutrients, and facilitating microbial decomposition, they enhanced the productivity and stability of benthic ecosystems. Their burrowing activity allowed protists, early mollusks, arthropods, and other multicellular organisms to access nutrients more efficiently, creating interdependent networks of life beneath the ice. In this sense, worms were not merely consumers, but ecosystem engineers, shaping the very foundations of life during Snowball Earth. The evolutionary significance of early worms lies in their combination of mobility, feeding versatility, and sediment engineering. By exploiting detritus, microbial mats, and small invertebrates, they maintained energy flow and ecological connectivity in extreme environments. Their burrowing behavior promoted the stability and productivity of benthic habitats, ensuring that life could persist in a world dominated by ice and extreme cold. Through these adaptations, worms demonstrated how soft-bodied, modular organisms could endure environmental extremes while shaping the ecological landscape for themselves and other species. Early worms, the burrowing engineers of Snowball Earth, exemplify resilience, functional innovation, and ecological influence. They moved through sediments, grazed detritus, redistributed nutrients, and facilitated the persistence of microbial and multicellular life. Through their activity, they connected microhabitats, enhanced energy flow, and maintained ecosystem stability in a frozen world. Their presence highlights the crucial role of small, flexible, and adaptable organisms in sustaining life under extreme conditions, setting the stage for the post-glacial diversification of more complex animals and the establishment of dynamic benthic communities. Early brachiopods, the shelled filter feeders of Snowball Earth. Amid the frozen, nutrient-scarce oceans of Snowball Earth, early brachiopods emerged as one of the first groups of shelled, filter-feeding animals. Though often overshadowed by their mollusk cousins in evolutionary studies, brachiopods played a critical role in structuring benthic ecosystems, stabilizing sediments, and contributing to nutrient cycling. Their unique combination of rigid shells, feeding structures, and sessile lifestyle allowed them to thrive beneath thick ice sheets where light was limited and energy resources were patchily distributed. One of the most significant adaptations of early brachiopods was their mineralized bivalve shells, which provided protection from predators, physical stress, and harsh environmental conditions. Unlike mollusks, whose shells often incorporated mobility and burrowing strategies, brachiopod shells were designed for a sedentary existence. Anchored to substrates via a stalk-like pedicle, these animals could stabilize themselves in turbulent sub-ice waters, resisting currents and ice movements while maintaining access to nutrient-rich water flows. The shells not only offered defense, but also helped maintain internal fluid balance and supported the delicate feeding apparatus inside. The feeding system of early brachiopods was specialized and highly efficient for the low-energy environment of Snowball Earth. Using a lophophore, a crown of ciliated tentacles, they generated water currents to capture suspended particles, including bacteria, protists, and detritus. This passive filter feeding strategy allowed brachiopods to exploit food resources that drifted past them, conserving energy in a world where actively hunting prey was often impractical. The structure of the lophophore, with its dense ciliation and large surface area, maximized particle capture even in low-flow conditions beneath the ice. Early brachiopods were highly versatile in microhabitat selection. While some colonized hard substrates like rocks, microbial mats, or shells of other organisms, others settled in softer sediments where currents provided a continuous supply of suspended nutrients. Their sessile nature allowed them to maintain consistent access to waterborne resources without expending energy on locomotion. Some may have formed small aggregations or colonies, increasing feeding efficiency by creating local water turbulence and enhancing particle capture. These microhabitat choices were critical to survival in a fragmented and extreme environment where patches of habitable space were limited. Reproduction in early brachiopods contributed to their persistence under extreme cryogenian conditions. They produced eggs capable of withstanding low temperatures, low oxygen, and prolonged periods of starvation. Free-swimming larvae allowed dispersal across benthic habitats, ensuring colonization of available niches and maintaining genetic diversity. Some species may have reproduced asexually through budding or fragmentation, allowing local populations to recover after physical disruption or ice-induced mortality. These reproductive strategies provided both short-term resilience and long-term evolutionary continuity, enabling brachiopods to remain integral components of subice ecosystems. Brachiopods also integrated into early ecosystems through interactions with other organisms. Their shells provided surfaces for microbial colonization, contributing to localized productivity and habitat complexity. By filtering water, they facilitated nutrient cycling and influenced the abundance and distribution of microorganisms, including bacteria, algae, and protists. Small motile organisms, including arthropods and microscopic worms, may have used brachiopod shells as shelter or feeding grounds, further increasing the ecological interconnectedness of Snowball Earth communities. The fossil record provides evidence that brachiopods were present during the cryogenian glaciations, though their delicate bodies often left only sparse impressions or shell fragments. These fossils indicate that early brachiopods were not only surviving, but establishing functional communities anchored to substrates and exploiting water flow for filter feeding. Despite the extreme cold, low light, and patchy nutrient availability, brachiopods maintained consistent activity, contributing to sediment stabilization and supporting other benthic organisms in microhabitats. Modern analogues provide insight into the survival strategies of cryogenian brachiopods. Contemporary cold-water brachiopods, including polar and subglacial species, survive near freezing temperatures, low oxygen levels, and limited nutrient availability while maintaining filter feeding activity. Many rely on slow metabolic rates, energy-efficient feeding, and symbiotic microbial associations, strategies likely shared with their cryogenian ancestors. Observing these living species demonstrates how early brachiopods could persist in extreme environments while remaining integral to ecosystem function. Brachiopods' ecological contributions extended beyond individual survival. By filtering water, they regulated microbial populations, facilitated nutrient redistribution, and stabilized benthic sediments. Their shells created structural complexity in otherwise homogeneous subice environments, providing surfaces for colonization by microorganisms and small multicellular animals. Aggregations of brachiopods may have enhanced local food capture efficiency and maintained ecological balance, demonstrating that even sedentary organisms could shape ecosystem structure significantly. The combination of mineralized shells, efficient filter feeding, and reproductive versatility exemplifies the adaptive ingenuity of early brachiopods. These features allowed them to survive prolonged glaciation periods, persist in fragmented habitats, and interact with other organisms in ways that stabilized and enhanced benthic ecosystems. Brachiopods were an essential component of Snowball Earth communities, providing structure, nutrient cycling, and energy flow that benefited both microbial and multicellular life. Early brachiopods, the shelled filter feeders of Snowball Earth, highlight the significance of structural innovation combined with ecological integration. Anchored to substrates, capturing waterborne nutrients, and facilitating microhabitat complexity, they contributed to the resilience and organization of frozen ocean ecosystems. Their adaptations illustrate how multicellular life could endure extreme conditions through energy-efficient strategies, symbiotic relationships, and environmental integration, ensuring the persistence of life beneath ice-covered oceans and paving the way for post-glacial diversification. Early bryozoans, the colonial builders of Snowball Earth. Beneath the thick ice of Snowball Earth, life found ingenious ways to persist in extreme conditions, and among the most fascinating survivors were early bryozoans. These tiny, colonial animals may have been only millimeters in individual size, but their ability to form interconnected colonies allowed them to establish a presence in frozen oceans, stabilize benthic habitats, and create micro-ecosystems that supported other life forms. Their survival demonstrates how cooperation and modularity can be a powerful evolutionary strategy in environments characterized by scarcity, turbulence, and low light. One of the key adaptations of early bryozoans was their colonial lifestyle. Individual zooids were physically connected through a shared tissue matrix, which allowed resources such as nutrients and energy to be distributed throughout the colony. In a harsh environment like Snowball Earth, where local nutrient patches could be sparse and patchily distributed, this integration meant that polyps capturing food in one area could share energy with less fortunate or damaged members of the colony. This strategy increased the resilience of the group and ensured the persistence of the organism even when individual zooids faced localized stress or predation. Bryozoans were also structural builders. Their colonies grew on rocks, microbial mats, shells, and other stable surfaces, forming branching or encrusting patterns that enhanced habitat complexity. These structures stabilized sediments against ice-induced currents and turbulence, creating safe spaces for microorganisms, small worms, and juvenile mollusks. The colonies acted as living scaffolds, increasing the availability of attachment sites for other organisms and creating localized refuges in otherwise harsh benthic environments. By forming these interconnected frameworks, bryozoans played an important role in shaping microhabitats and increasing the ecological connectivity of subice ecosystems. Feeding in bryozoans was highly specialized. Each zooid possessed a lophophore, a crown of ciliated tentacles used to generate water currents and capture suspended particles, including bacteria, protists, and detritus. The shared energy network within the colony allowed individual zooids to be efficient even in low food conditions, as captured nutrients could be transported across the colony to support growth and reproduction. This passive filter feeding strategy conserved energy in an environment where active hunting was often impractical and enabled bryozoans to exploit patchy food resources distributed by currents beneath the ice. The morphology of bryozoan colonies varied widely. Some formed encrusting sheets, tightly adhering to surfaces and resisting the mechanical stresses of ice movement and currents. Others developed branching or tree-like forms, extending their lophophores into the water column to capture more food, while simultaneously increasing the structural complexity of the benthic habitat. These growth forms not only enhanced feeding efficiency, but also created niches for smaller organisms, influencing community composition and facilitating symbiotic interactions within Snowball Earth ecosystems. Reproductive strategies in bryozoans further enhanced survival. Colonies could reproduce asexually through budding, expanding their presence across favorable surfaces. Sexual reproduction produced larvae capable of dispersal through water or sediment, colonizing distant microhabitats and maintaining genetic diversity. These larvae may have entered dormant states to survive freezing temperatures or nutrient scarcity until suitable substrates or environmental conditions were encountered. This combination of reproductive strategies allowed bryozoans to persist across fragmented habitats and endure the fluctuating conditions of a frozen world. Bryozoans also engaged in symbiotic relationships. Microbes, algae, and even tiny invertebrates could colonize the surfaces of the colonies, benefiting from nutrient accumulation and shelter, while bryozoans themselves may have hosted photosynthetic microorganisms in areas where thin ice allowed some sunlight penetration. This dual strategy of direct filter feeding and symbiosis provided an advantage in environments with low and inconsistent energy input, ensuring that colonies could thrive despite extreme resource limitations. The fossil record indicates that bryozoans were already present during the Cryogenian, though preservation is often limited to impressions or mineralized fragments. These fossils reveal branching, encrusting, and layered colony morphologies, demonstrating their adaptability in both form and function. Even in small numbers, bryozoan colonies contributed to the stabilization of benthic habitats, the creation of attachment surfaces, and the maintenance of nutrient flow, highlighting their role as key ecosystem engineers beneath the ice. Modern analogues in polar and subglacial habitats provide insight into the persistent strategies of early bryozoans. Contemporary bryozoans survive near freezing temperatures, low oxygen levels, and limited food availability while forming encrusting and branching colonies that stabilize sediments and provide habitats for other small organisms. Observing these living relatives suggests that Cryogenian bryozoans employed similar strategies, integrating modularity, structural complexity, and efficient energy capture to maintain colonies in extreme conditions. Their persistence emphasizes the importance of small-scale ecosystem engineering for the survival of life under harsh environmental stress. The ecological impact of bryozoans extended beyond individual colony survival. By stabilizing surfaces, creating habitats, and capturing suspended nutrients, they regulated microbial communities and provided safe zones for other multicellular organisms. Their presence enhanced ecosystem complexity, increased the flow of energy and nutrients, and contributed to the resilience of Snowball Earth benthic ecosystems. Even in fragmented, low-light, and ice-dominated environments, bryozoans maintained ecological networks and facilitated the persistence of diverse life forms. The evolutionary significance of early bryozoans lies in the combination of colonial living, structural innovation, and ecological integration. By forming interconnected colonies, these animals enhanced survival, optimized feeding efficiency, and created habitats that supported a wider range of organisms. Their strategies illustrate how modularity, cooperation, and ecosystem engineering can enable life to persist in extreme conditions. Through these adaptations, bryozoans became essential contributors to the stability and productivity of frozen ocean ecosystems, preparing the groundwork for more complex communities after the glaciations. Early bryozoans, the colonial builders of Snowball Earth, exemplify resilience, adaptability, and ecological influence. By forming interconnected colonies, capturing nutrients efficiently, stabilizing sediments, and providing microhabitats for other organisms, they maintained ecosystem function under conditions that challenged most life forms. Their strategies reveal how cooperation, modularity, and environmental integration allowed small, seemingly fragile animals to shape the structure, stability, and continuity of life beneath a planet-wide ice sheet. Early nematodes, the microscopic survivors of Snowball Earth. Beneath the global ice sheets of Snowball Earth, where sunlight barely penetrated and energy resources were patchily distributed, one of the most resilient and underestimated groups of organisms thrived: early nematodes. These microscopic, unsegmented roundworms were among the smallest multicellular animals in the Cryogenian oceans, yet their ecological influence was enormous. Their ability to exploit bacterial biofilms, sedimentary detritus, and microbial mats made them critical players in benthic ecosystems, linking energy flow from microbes to larger animals and maintaining the continuity of life in an environment dominated by ice. Early nematodes were remarkably versatile in their feeding strategies. Unlike the larger annelid worms that burrowed through sediment, Nematodes could inhabit the smallest interstitial spaces between grains, within microbial mats, or even inside subglacial brine channels. They fed on bacteria, archaea, protists, and decomposing organic material, extracting nutrients from sources that were inaccessible to larger organisms. Some species may have been opportunistic predators, preying on smaller nematodes or protozoans, while others were strict detritivores, scraping organic matter from sediment surfaces. This versatility allowed nematodes to persist in nearly every microhabitat beneath the ice, exploiting energy wherever it could be found. The small size and simple body plan of nematodes were key to their survival. Their elongated, flexible bodies, devoid of segmentation, allowed them to move easily through tight spaces in sediment or microbial mats. They possessed a complete digestive tract, with a mouth, pharynx, intestine, and anus, enabling efficient nutrient extraction. The simplicity of their physiology allowed them to tolerate extreme conditions, including low oxygen, high salinity, and near-freezing temperatures. Nematodes could slow their metabolism during periods of scarcity, entering states of dormancy that preserved energy and increased survival rates over extended intervals when food was limited. Reproduction in early nematodes was highly adaptive. Many species were capable of both sexual and asexual reproduction, allowing them to rapidly expand populations when conditions were favorable while maintaining genetic diversity. Eggs and juvenile stages could survive freezing, desiccation, and low nutrient conditions, enabling dispersal across fragmented habitats. Some species exhibited parthenogenesis, producing viable offspring without fertilization, a strategy that ensured population persistence in isolated or extreme microenvironments. This combination of reproductive strategies allowed nematodes to maintain high population densities, even in habitats with sparse food and limited space. The ecological role of nematodes extended far beyond their individual survival. By feeding on bacteria and detritus, they regulated microbial populations, preventing the overgrowth of any single species and maintaining ecological balance. Their movement through sediments and microbial mats redistributed organic material, facilitating nutrient cycling and oxygen penetration. These microscopic engineers created microzones of enrichment that supported other small invertebrates, protozoans, and juvenile stages of larger animals. In this way, nematodes connected primary producers and detrital communities to higher trophic levels, serving as critical links in cryogenian food webs. Nematodes also displayed remarkable tolerance to environmental extremes. Their bodies could survive periods of desiccation or freezing, and some species tolerated hypersaline or low oxygen microenvironments within sediments or brine channels. This resilience allowed them to persist in microhabitats that were uninhabitable for most other animals. By occupying these niches, nematodes ensured the continuity of energy flow in sub-ice ecosystems, maintaining the availability of prey for larger organisms, including early arthropods, mollusks, and worms. Their ability to endure extreme conditions highlights the evolutionary advantage of small size, simple physiology, and metabolic flexibility in the cryogenian oceans. Fossil evidence for nematodes during Snowball Earth is limited due to their microscopic size and soft bodies, but trace fossils, burrows, and preserved microbial mat interactions suggest their widespread presence. These microfossils indicate that nematodes were abundant in sediments, actively feeding, moving, and influencing microbial composition. The density and distribution of nematodes would have been critical for maintaining ecosystem stability, particularly in habitats dominated by microbial life. where small-scale bioturbation and nutrient redistribution ensured the persistence of multiple trophic levels. Modern analogues provide insight into the survival strategies of early nematodes. Contemporary species living in polar regions, subglacial lakes, and deep-sea sediments demonstrate extraordinary tolerance to freezing, hypoxia, and low nutrient conditions. They employ dormancy, efficient feeding, and rapid reproductive cycles to persist in habitats that are highly fragmented or subject to extreme environmental stress. Observing these living nematodes suggests that cryogenian species employed similar strategies, exploiting microbial energy sources, navigating sediments, and maintaining ecosystem connectivity under conditions of global glaciation. The evolutionary significance of early nematodes lies in their combination of small size, metabolic efficiency, and ecological versatility. By occupying microhabitats, consuming microbial and detrital resources, and redistributing nutrients, they maintain the foundation of benthic food webs. Nematodes linked microbial productivity to larger animals, supporting complex ecological interactions even under the harshest conditions. Their survival demonstrates how microscopic organisms can have disproportionate influence on ecosystem stability, energy flow, and the persistence of life during extreme global events. Early nematodes, the microscopic survivors of Snowball Earth, exemplify resilience, adaptability, and ecological importance. By inhabiting sediments, consuming bacteria and detritus, redistributing nutrients, and supporting microhabitat complexity, they maintained the continuity of benthic ecosystems beneath the ice. Their strategies reveal the evolutionary advantages of small size, physiological flexibility, and ecological integration, illustrating how microscopic life could persist through extreme glaciations and sustain the foundation for the diversification of more complex animals in the post-glacial world. The creatures that survived the Snowball Earth era show that life finds a way even in the harshest conditions. Every ice age, every frozen ocean, and every global catastrophe forced evolution to invent new strategies, discard old ones, and rewrite the rules of survival. From microscopic nematodes and early arthropods to shelled brachiopods, colonial bryozoans, and filter-feeding cnidarians, each lineage that endured helped shape the next wave of life. Please like, subscribe, and join us for the next video if you enjoyed this journey into the frozen oceans of deep time. The world before the dinosaurs is filled with stories more extraordinary than most of us ever imagined.